Thank you. Okay. So I'm joining CGI array tomography team. Also, I'm doing 3-view serial block phase, volume microscope, and then I'm doing correlative microscope. But today, thank you, Liu Chin, giving me this opportunity to talk about the freeze fracture replica immunogold labeling. I love this technology, but unfortunately, in the world right now, not many lab is using this technique. So I try to give you some idea what the freeze fracture replica can do, and then I hope everybody can be excited to consider to apply this technology for your research. Okay, this way. Maybe some of you remember you saw this type of figure in your old EM textbook. Freeze fracture EM was developed late 50s, and then it was used a lot in 70s. It's a really powerful tool, but basics is really, really simple. We can freeze a sample, and then we can fracture sample, and then we do deposit some evaporated material, heavy metal shadowing, such as platinum, and then carbon to make replicas. After making replicas, we can examine this replica as a structure of newly exposed surface. So that's why mostly this is for membrane analysis. But if we add etching process, we can see some of internal structure as well. So this is another view or more biological view of the concept of freeze fracture EM. We do freeze sample, and then we use frozen sample in a freeze fracture machine. And then we fracture the frozen sample. So fracture plane is going to often in between the repeat bilayer two leaves, because this is really physically weakest point of biology. So after fracturing, we can get two leaves of repeat bilayer. One is external phase called E phase, and then one is protoplasmic phase, the freeze fracture protoplasmic phase P phase. So, but usually, or not usually, original technology, freeze fracture EM, was using very strong chemicals such as bleach to digest all biology below the replicas. So there was no chance to do immunolabeling. So that was that is one of the reasons this freeze fracture technology is not used anymore. But not now, since 1995, Professor Kazushi Fujimoto developed this technology to advance, using SDS as a digestion material to wash out the biology from replica, and then that allows us to label the object on the top of freeze fracture replica information. So this was really a big breakthrough. Now we can see freeze fracture image and some composition information from this immunolabeling. So this is his original picture of SDS freeze fracture replica labeling, SDS-FRL. So he used this very unique and then very famous specific membrane structure, such as tight junction or gap junction. And then you can see this very typical structure view, tight junction string or plug of gap junction with these deposit gold particle dots. So after this, doing this, we can really identify the pure, not only the pure morphology, how this structure is making arrangement, but we can also see the chemical composition by immunolabeling. I was really lucky to learn this technology from him originally, But after he sent me to John Rush's lab in Colorado State University, he passed away at the age of 49. So speaking of tight junction, now we have all knowledge about how tight junction looks like on a replica business. They are making very complicated string structure. But now we have to think about if we only use thin section TEM. Maybe I should use this one, laser pointer. Yes, so if we use this type, we see the tight junction in a thin section EM. These contacting points are really the tight junction, communicating between two cells. So probably or almost exclusively, it's impossible from reconstruct the tight junction strand structure only from thin section image. So now let me put back my original title slide picture. You can see the freeze fracture image, and then I colored something. But this side, we have cross fracture of cytoplasm, and then you can see some synaptic vesicle. And then we have here two different phases of cell membrane. Here we have extrasynaptic E phase of postsynaptic cell. And then this blue portion is P phase, protoplasmic phase of
I was
synaptic
removing
cell.
this one and moving this in case. I was putting on in my.
Okay. And then yes, I hear this blue is P phase, and then here there are small bumps you can see. These are actually integral or intramembrane protein IMPs. Some of those are really sparsely localizing on the membrane, and then some of those are making this cluster or aggregation. If I take off the color, you can see those structure better. Here, some IMPs are making these groups. And then on this P phase, you can see better, we have now Immunogold labeling. This replica has two sizes of gold particle. So small gold particle was used for labeling GFP. GFP was used for a marker of specific cell type. And then this large gold particle is indicating the localization of calcium channel for this case. And we are doing neuroscience, so we often need to see or analyze synapse. Synapse is a communication place between pre- and postsynaptic cell. And then we have this particular structure. For this replica image shows the presynaptic side as a cross fracture of cytoplasm, and then you can see synaptic vesicles. And then postsynaptic side is shown in E phase, and then we have this particular unique view of IMP cluster. And then we have these gold particles. This is a labeling result of AMPA type glutamate receptor. So from this view, we can see the glutamate receptor containing IMPs are really making this arrangement on the postsynaptic side. But again, if we think about thin section as a comparison, in the thin section EM, we have synapse, of course, and then this is a very typical view. The opposing side of presynaptic, postsynaptic cell, we have synaptic vesicle in a presynaptic view. And then postsynaptic side, we have this electron dense material here. This is postsynaptic density. And then we can see these two membranes facing each other. But this receptor protein is really lining on the postsynaptic membrane. So again, this is actually the better view when we want to analyze membrane protein distribution or arrangement, because we have a 2D view here. So let's go to a little bit more detail how this freeze fracture replica immunogold labeling works. We first anyway freeze sample, and then we fracture sample, and then we have exposed the membrane, and then we do make a shadow, and then carbon coating to make replica more rigid. After that, we do use SDS solution to digest almost all of biology. But this is a trick. We can leave a little bit biology below the replica. Then we have epitope remaining here. So that's why we can do primary antibody, secondary antibody reaction to the replica, just like other EM technology, immuno-EM technology. Then we can examine replicas in TEM. As a result of that, we can see something like this. For this case, we used, again, the large particle for channel, and then small particle for the cell marker GFP. So we see something like this. So speaking of freezing, this is really your option to fit to your experimental designs. We can use both rapid freezing or high pressure freezing, and then sample could be fresh or fixed, or with or without cryoprotection. And we can choose how to freeze sample depends on your purpose. If sample is small enough or freezing depth is narrow enough, we can go plunge freezing or metal contact rapid freezing or high pressure freezing. For our case, mostly we want to use high pressure freezing because we are handling the tissue, and then we want to get large enough object to examine. So from here, I want to show how to make replica using our machine. We are using this JFD2 equipment from JOL. So we have two different type of table. Actually, more if you want, we can make a customized table to fit to your sample. So this is a very standard single replica table. So sample has to be sticking out from this, and then we do knife cut of the sample. And then for the case of double replica, we can use this type of sandwich holding table. This is a sample rod. We can insert the sample to the machine. And this double replica table is now screwed in. So now here, Debbie is making replica. And then first we do insert frozen sample from liquid nitrogen quickly to the freeze fracture machine. The first machine is pre-vacuuming this sample chamber. And then when it reaches to the high vacuum condition, we hear the sound changed, and then we open the valve and then insert the sample. After inserting sample, we can just set the table to the center of this rotation table. After inserting sample, actually this is a demo, it's not a freezing condition, but you can see we put this knife inside of this shroud. So for the case of double replica, we do use this knife edge to open double replica table. And then as a result of that, our sandwiched sample is opened, and then we can have both sides as a replica. When we do knife fracturing, we do use actual knife portion. Immediately after opening or exposing sample, we do first rotate sample to coat the precarbon. And then here you can see this actual evaporation. By adding filament current, this material starts evaporating. And then we have shutter here. And then by opening shutter, we have deposition of material on the top of exposed surface. After making replica, we can take out these samples from the machine. This is how it looks like. you can see this slightly gray color on the top of tissue pieces. So this is a result of deposition of platinum shadow and then carbon. From here, we use this type of platinum loop or wire to take out these pieces and then put it into the SDS solution and then digest at the higher temperature. After digestion, we can do immunolabeling, starting from blocking, then primary, secondary antibody. As you see, this experimental condition is really variable because this is totally depend on your epitope. Some epitope is exposed easier or harder, and then for the immunolabeling, we need really the optimization. But one thing I wanna ask your attention is here, for particularly this purpose, we chose this type of temperature and then thickness of replica. This is, so before going that, yes, this is after doing this replication, we do pick up replicas on EM grid. So this is 3 millimeter regular EM grid. So you can imagine how big replica could be. So this is one example to show why this deposition condition is important. Then also this is depend on the fracture temperature. This is only 1 gig slide, today's talk. So this is just the example. This is a stereoscopic view of aquaporin 4 square array. A and B are actually from same structure of aquaporin square array water channel. But what's the difference is freezing temperature and then coating thickness. So this top view has this more globular looking of this square array. But actually now compared to this B, you can imagine this A is like overcoated. So this tells us really that we have to always consider free fracture temperature, and then pre-carbon coating is probably necessary when we see something tiny. And then also this material coating temperature is also important. I don't want to go too deep, but there are lots of optimization needed to get the best information for your purpose. So this is also another frequently asked question. How many different protein can be detected from one replica? So far, we could successfully did 4 different epitope detection because, of course, we have to use gold particle for the marker for this case, and then we use the 6, 12, 18, 14, 40 nanometer gold particle. This is doable, but sometimes because we are using commercially available secondary antibody gold conjugated, batch by batch, we sometimes has the size variability of individual gold particle size. So occasionally we have some uncertainty to distinguish if this gold particle is a 6 or 12 or 12 or 18. But if we have a good batch of gold conjugated secondary, it could be possible. So now we have labeled the replica. So what do we want to do? Of course, we want to analyze and to extract biological information from replica, labeled replica view. One good thing of replica labeling is really high labeling efficiency, because I explained this replica technique is using SDS digestion. So it's just like SDS page. So you can imagine this target molecule is denaturing below the replica. So it makes antibody access much easier. So we can get the really highest labeling efficiency among immuno-EM technology. Then also we have really 2D view as we were talking about. So we can really see arrangement of this membrane protein. But also we have to remember always when we are using immunoreaction, this is immunoreaction, and then we don't know how many epitopes are occupied by antibody or how many antibodies actually touching to the epitope. This is the same idea with other immuno-EM technique. For example, what kind of information we can extract from this type of image? We often want to understand this type of synapse size or density of gold particle, or if they are mixing or co-localizing, two proteins are co-localizing, what is the ratio of those? So for this case, we have to demarcate the area of interest and then count the gold particle. So if it's really more wide range of labeling like this, and then we want to know this positional relationship of, for this case, channel. How these channels are distributing, they are close to each other, or they are making a cluster, and these clusters are how close these clusters are. These are another information to extract biological information from this distribution. For this case, we have to really get the spatial coordinates, means all XY coordinates of all these gold particles, and then we can somehow extract these relationships. So I'm saying extract XY coordinates, how to do it. The first thing is you see this tiny, tiny slow process here. This is actually our manual counting. This is the most simplest way. And the manual annotation works, but you can imagine it's time consuming, laborious. If this is only one image we want to analyze, no problem. But usually we have to analyze hundreds or maybe thousands of images. So we need a good technology to do this for us. Ideally, fully automated. So one thing we have been studying is really deep learning technology recently. Some of image analysis software integrated already this deep learning module in their user interface. So this is an example from Dragonfly ORS. By training the network, we can really efficiently extract these gold particle information. For this case, we put the mask on it, and then network is extracting gold particle from this individual mask. And then you can see red and blue color. These are small gold, large gold together. So we can get this extraction, and then of course software can output all XY coordinates for us. But then what we can do? We have now all XY coordinates of gold particles, and then how to analyze. Again, this could be manual, but we developed this really helpful tool. This is done with Seth Godwin. He was our high school intern, actually, originally, when he started working with us. And then he wrote this program, and then we published in this paper. And then this program is actually open through Max Planck GitHub. What we can do is, this is a user interface of this program, Gold In and Out, GIO. We can upload our image and then XY coordinates, and then we can select what we want to analyze, workflow, and then just to start. And then we can get such as nearest neighbor distance or clustering or cluster separation, these things. And then sometimes in our field, we want to know gold particle distribution relative to our biological landmark. So we developed also this type of stuff. And then good thing is this program is always comparing real distribution to the random distribution. So we can get both information together. This is some example of output, nearest neighbor distance or cluster separation. These are actual output from this GIO program. So today I talked about this freeze fracture replica Immunogold labeling method, how to make replicas, to how to analyze replicas. I hope this gives you some idea. Maybe you want to do something, and then I think we can do it because I know lots of old freeze fracture machines are sleeping in the lab. So we can use it, and then this replica technology gives us some new insights of your research. Of course, some skill set is needed, but our lab is really happy to help your endeavor. So please contact me if you want to try. And then, yes, like integration of deep learning analysis, we can extract lots of data from the replica images. And this, again, GIO is also available for everybody if you want to try. At the end, I want to thank you to my team, electron microscopy core, and then Max Planck is having actually imaging center, both light microscopy, electron microscopy. We are working to provide application support for not only internal people, but we have lots of external collaboration as well. And then I also want to thank you for my collaborator, current or former group leaders from Max Planck. And then finally, I want to thank you to JOL Japan. This machine is still supported by Japanese Akishima team, and this is a great machine. And then I hope this technology is going to be used more. Thank you so much. And then I'm ready to answer your question.
Okay, any question for Naomi? Okay, Naomi, so you talk about the SDS digest. So how does it happen that you will over digest, actually lose the antigen? How do you know how to optimize the time that required to get the optimal digestion?
That's a good question. And then maybe you know the answer because we have to try, like a short digestion, long digestion, and if it's over digestion, of course, we don't have any labeling. And then if we less digestion, we have some protein debris remain on the replica. So we have some like a smudgy protein material, and then gold particles are trapped. So it's sort of a balance between those. But generally saying the membrane integrated protein, for example, we can use 80 degree. And then if it's a membrane protein binding protein, we can reduce the digestion temperature, like 60 degree. So this is another option we can think from the position of our target protein, what kind of condition we can use.
Okay.